26th question. On stem, an epidermal hair. These epidermal hairs are present mostly on dicot stem. They are present on dicot stem epidermis. This is the outermost covering of dicot stem. This is outermost covering of dicot stem known as epidermis. This is called epidermis. And it is provided with the cuticle. This is cuticle. Cuticle is not a cellular structure. It is not a cellular structure. It's a deposition of cutin only. It's a deposition of cutin only. And on dicot stem you see multi-celled stem hair. This is multi-celled stem hair. As appendage. It is not the outgrowth of epidermal cell. It is present as appendage over the epiderm. And any epidermal appendage is called trichome. Any epidermal appendage is known as trichome. That's why these are also trichome. So the answer of 26th question is trichome. Answer of 26th question is trichome. And these two structures not concerned with the botany, but trichoscleride is concerned with the botany. Trichoscleride is type of a scleride which is fine and hair-like. Fine hair-like scleride is known as trichoscleride. Fine hair-like scleride is called trichoscleride. So the answer of this question is trichome only. Epidermal hair as appendages. They are not the outgrowth of epidermal cell as in root hair. Root hair is actually outgrowth of epidermal cell. It is not the appendage. Then bulliform cells are present. First, these bulliform cells are concerned with the leaf. Bulliform cells, they are found in leaf. They are associated with the epidermis of leaf. Bulliform cells are found in leaf. They are associated with the leaf. And especially monocot leaf, not in dicot. They are in monocot leaf and upper epidermis, because leaf has two epiderma. They are found in upper epidermis. They are found in upper epidermis, and for upper we use the word adaxial. We use the word adaxial epidermis. Adaxial epidermis. So this is epiderm of a monocot leaf, upper epidermis of monocot leaf, and you see few large and empty cells. Few large cells. These are the few large cells associated with the upper epiderm of leaf. These are bulliform cells. These are bulliform cells. They are also called motor cells. These are also called motor cells. Bulliform cells are also known as motor cell. And especially in grasses. They are found in especially in grasses. Monocots, that is grasses are included in the monocot. When these cells lose their turgidity, the leaf curl inward. When they gain the turgidity, there is an uncurling of leaf. Uncurling or unfolding of leaf. That is, folding and unfolding of leaf is related with the bulliform cell, and it is also a Turger operated phenomena. It is also related with the or we can say it is a turgor operated phenomena. When they curl, they prevent the loss of water. When they curl, they prevent the loss of water. So here the answer of this question is, that is, they are found in adaxial, that is upper surface, upper epidermis of grasses, and role I have already described. 28 question. Which of the following structure is not completely? Underline keywords. One is not, then completely parenchymatous in dicot stem. We take the cross section of dicot stem. This is epidermis. Outermost covering is epidermis. It is with cuticle, that is with cuticle, followed by cortex is there. Then cortex is present. And cortex ends with the endodermis. This is the innermost layer of cortex, that is endodermis. This is endodermis. And these are rich in starch grain. That's why endodermis is also called a starchy sheath in dicot stem. This is the endodermis of dicot stem. Just below the endodermis, pericycle is present. Just below the endodermis, pericycle is present. And see the pericycle. This pericycle is in patches. This pericycle is in patches. That is, sclerenchyma and parenchyma are alternately arranged. Sclerenchyma and parenchyma, they are alternately arranged. This is a sclerenchyma. These are all sclerenchyma. The sclerenchyma and parenchyma, they are alternately arranged. Sclerenchyma is dead. This is sclerenchymatous portion. This is parenchymatous portion. I mean, not completely parenchymatous. Not completely parenchymatous. In root, it is completely parenchymatous. In monocot stem, it is not found. Pericycle is not there in the monocot stem. In leaf, it is not found. But in dicot stem, pericycle is there. Pericycle is present, and it is just below the endodermis. Just below the endodermis, and see, it has sclerenchymatous and parenchymatous patches alternately arranged. So the answer is pericycle. The answer is pericycle. It is a part of stele. Stele is central vascular cylinder. Stele is central vascular cylinder. And this is the structure of vascular region. And see, xylem and phloem are alternately arranged. Xylem and phloem are alternately arranged. That is, first of all, these are the radial vascular bundle. These are the radial vascular bundle. These are the radial vascular bundle. And radial vascular bundles are found in root. Second, if the vascular bundles in root are more than 6, then it is monocot root. Then it is monocot root. Less than 6, dicot root. See this one. This is one bundle, one xylem, one phloem, one bundle. This is bundle number 2. This is bundle number 3. This is bundle number 4. Bundle number 5. Bundle number 6. Bundle number 7. Bundle number 8. Generally in monocot root, radial vascular bundles are 8 to 32 in number. 8 to 32. That's why finally, number 1, these are the radial vascular bundle. So no chance of leaf, no chance of a stem. But now 2 options are left, that is radial, root and dicot and monocot. But see the number. If number is more, then monocot. If the number is less, then they are dicot. So final answer, this is monocot root. Second identification, this is large pith. Large pith is found in monocot root, whereas it is small in dicot root. Pith is small in dicot root. Identify the statement true or false. Conjunctive tissue. Conjunctive tissue is parenchymatous, lie between xylem and phloem and dicot root. It is right. Vascular bundles are eustelic in vascular bundles are eustelic in dicot stem. Right. Both are right. What is conjunctive tissue? That is tissue present in between xylem and phloem. See the past picture. The tissue present in between these two, that is xylem and phloem. This is xylem and phloem, the space between them. Here the conjunctive tissue is present. Here the conjunctive tissue is present. Let's clear then explain. What is conjunctive tissue? Conjunctive tissue is present in between xylem and phloem. That is in this location. This is the location of conjunctive tissue. That is parenchymatous. This is conjunctive tissue and parenchymatous. Conjunctive tissue made up of parenchyma. Made up of parenchyma. So next question, that is 30th question. That is about the conjunctive tissue. And what is eustele? Eustele is well organized stele. Eustele is well organized stele. And what is well organized stele? That is, it has pericycle. Eustele consists of pericycle. Number 2, vascular bundles, and they are in ring. They are in ring. Then substantial pith is present. Pith, that is medulla. Pith, that is medulla. And pith rays, that is medullary rays. These all 4 components are found in eustele only. And eustele is present only in dicot stem, not found in any others. Not found in any others. For information, atactostele is found in monocot. Stele of monocot stem is atacto. Atactostele. As against in dicot stem, eustele is there. In monocot stem, atactostele found in monocot stem. That is unorganized type of stele. Atactostele is actually unorganized, whereas eustele is well organized stele. Well organized stele. So both the statement here given, they are true. They are true. 31st question. Starchy sheath. In dicot stem, when we take the study in dicot stem endodermis. Endodermis of dicot stem, rich in starch grain. Rich in starch grain. These are all starch grains. They are abundantly found in cells of endodermis. Cells of endodermis of dicot stem. That's why their endodermis is known as, their endodermis is called starchy sheath. So the answer is the first one, that is endodermis, that stores starch grain, and in dicot stem only. Dicot stem only. Not applicable for other endodermis. Dicot stem. Casparian strips are found in root. Casparian strips are found in roots of dicot as well as in monocot. But endodermis in the form of starchy sheath found only in dicot stem. Found only in dicot stem. Then palisade parenchyma. In dicot leaf, the tissue present between two epidermis is mesophyll tissue. This is upper epidermis. This is upper epidermis or adaxial epidermis, and this is the cuticle. This is lower epidermis. This is lower epidermis. It is also provided with the cuticle. This is cuticle. And in between two epidermis, what is present? That is mesophyll tissue is present. The mesophyll is divisible in dicot leaf. Mesophyll is divisible in dicot leaf. Towards upper epidermis, cells are compactly arranged. Cells of mesophyll are compactly arranged. They are chlorenchymatous mesophyll cells. They are compactly arranged. Whereas towards lower epidermis, you see they are arranged with large air spaces. Clear cut difference between the two mesophylls. That is, in monocot leaf, mesophyll is not differentiated, it is differentiated in dicot. Upper epidermis, where the mesophyll cells are compactly arranged, they are elongated cell arranged vertically. They are elongated cell arranged vertically. And towards adaxial epidermis, not abaxial. Towards adaxial epidermis, not abaxial. Such mesophyll tissue is called parenchymatous. Sorry, it is palisade mesophyll. Palisade mesophyll is upper side. That is, towards upper side it is palisade, and towards lower side it is spongy. Towards lower side it is spongy. So, mesophyll is divisible in dicot leaf. Upper side compactly arranged elongated cells, they form, that is, palisade mesophyll, and towards lower side they are loosely arranged with air spaces, that is known as spongy mesophyll. This is first half palisade and this is the second half, that is spongy. Arrange the following event according to their occurrence during the secondary growth. Yes. When secondary growth takes place? Secondary growth takes place in dicot stem and dicot root. Secondary growth takes place in dicot stem and dicot root, as well as in gymnosperm also, Secondary growth is due to lateral meristem. Secondary growth is due to lateral meristem, cambium and cork cambium. Secondary growth is due to lateral meristem, that is cambium and cork cambium. One is cambium, second is cork cambium. These tissue are responsible for the secondary growth. What is secondary growth? That is formation of secondary tissue by the activity of lateral meristem, due to which thickness increases, known as secondary growth. In dicot stem, there are two regions where the secondary growth is seen: within stele and outside the stele. Within stele and outside the stele. Within stele, let's see, vascular bundles are arranged in a ring. and in between xylem and phloem, primary xylem and primary phloem, a strip of cambium is seen. This is cambium. Prerequisite for the secondary growth in steel. Prerequisite for the secondary growth in steel is formation of cambium ring. And cambium ring formation requires another strip of cambium. Another strip of cambium which appears in medullary rays. This is seen only at the time of secondary growth. That is, medullary rays differentiates. Medullary rays undergo differentiation and form, and what is formed by them? That is, interfascicular cambium is formed. This is fascicular cambium, this is fascicular cambium, and in between them interfascicular cambium is there. So first one, for the formation of cambium ring, there is a differentiation of medullary rays. And after the completion of cambium ring, if cambium ring is formed, then both the side new tissues are formed. Towards inner side secondary xylem, towards outer side secondary phloem. Towards outer side secondary phloem is formed. So second event after the formation of cambium ring is formation of secondary xylem and secondary phloem. So if we arrange these one, to start with the second, then come to the first, that is second event. Then extrastellar secondary growth takes place due to phallogen or cork cambium. It is entirely secondary in origin. So for extrastellar secondary growth, third is required. If third is formed, then only cork cells are formed. Then only cork cells are formed. So the correct sequence with regards to the secondary growth in dicot stem: first, differentiation of medullary rays, that is second option is first one. Then formation of secondary xylem and phloem, this is second. Then cork cambium or phallogen is formed. Then only there is a possibility of the formation of cork cell. The second one is most appropriate with regards to the second, with regards to the sequence. This is the answer of 33rd question. Secondary xylem is called wood. Secondary xylem is known as wood. And after secondary growth, you see the TS of a dicot stem or a trunk, you see the maximum wood in there, that is still. And this wood is kept under two categories. The wood, secondary wood, is kept under two categories. Centrally located dark colored wood. Secondary located dark color wood is heartwood, is prominent one. That is secondary centrally located and prominent one is, it is dark colored, it is heartwood. It is what? Heartwood or duramen. It is heartwood or duramen. It is non-functional because vessels, they have tylosis. Vessels are with tylosis. And dark color is because of presence of tannin, resin, oil, gum in those cells. Then peripheral wood. This is peripheral light colored wood. This is peripheral light colored wood is sapwood or alburnum. Sapwood or alburnum. Sapwood is also called alburnum. Sapwood is also known as alburnum. It is light colored peripheral wood. Second, it is physiologically active. It is physiologically active, that is able to conduct water and minerals, because vessels are not blocked. Vessels are not blocked. So in this sense, in this case, dark colored is heartwood, central wood is heartwood, deposition of organic matter, all these three properties are concerned with the heartwood. The only one property which is left, that is peripheral light colored wood, is sapwood or alburnum. This is alburnum and is chief conducting part of xylem in case of secondary growth, after secondary growth. Then tyloses. What are the tyloses? Heartwood vessels. Heartwood vessels have balloon-like structure formed by the neighboring cells. Vessels of heartwood, they have balloon-like structure formed by the neighboring parenchyma and they contain metabolic waste. Such structure is known as tylosis. This is tylosis. So it's a balloon-like structure, outgrowths, parenchyma actually, it is outgrowth of parenchyma, and the lumen of vessels, present in the sieve tubes, wrong, temporary thickening, wrong, present in secondary xylem, this is correct, and makes it non-functional. In previous one, up to here is right, but tracheate cells are not involved in the formation of tylosis. Tracheidal cells are not responsible for the formation of these structure. Hence, the fourth one is correct. They are formed in the secondary xylem. They are balloon-like structure, but formed by the neighboring parenchyma. And because of these tyloses, secondary xylem is non-functional. That is not able to conduct water and mineral. So, out of these four options, fourth one is absolutely right. Right. These tyloses are seen in duramen, that is heartwood. In their vessels, these balloon-like structures formed by neighboring parenchyma. And duramen is non-functional, that is not conduct water and mineral. Duramen is non-functional. 36th question. All tissue exterior to the vascular cambium, that is known as bark. Non-technically, it is called bark. So, first one, that is A option is, that is first A. For A we can use the word bark. Then C, consist of. Bark is made up of. So, second, first one, third and fourth directly cancelled. Let us start with the bark, but it is not a living permanent tissue. This is again wrong. Then second option is correct. First one, it is bark and it is dedifferentiated and redifferentiated tissue. Dedifferentiation, that is, if a living permanent cell regain the power of division, that is dedifferentiation. And because of dedifferentiation, phallogen is formed. This phallogen forms new cell on both the sides. That is, towards outer side it forms scarp, towards inner side it forms secondary cortex. And they also lost the power of division. That is redifferentiation. Cells obtained from phallogen, they are redifferentiated cell. And phallogen itself is a dedifferentiated structure. That's why better option is second one. That is, better option is second one.